Okay. Well, it's more on the rest of the building. So
special needs. Great.
Oh, I thought we were gonna do the narrative.
Sorry, I just wanted to say hello. I emailed you. Okay, Gary, nice to meet you. I don't know if you have time to talk after. But yeah. Those are means I don't know, let someone go. So,
How's it going?
Okay.
Hi. Happy to be here. All right, I do know here.
So I'm actually a research associate now. Not making like monopoly money anymore. We just we wrote an engage grant together for about 8 months of funding. So the partner, it's up to 12. Okay,
That's one break.
sorry. Yeah, I mean, there's
Sit. I wish. Arch it. No, it doesn't.
Ray, I'm standing room only.
Oh, you know, it's cold.
Yeah, but it's Mercury.
last layer.
You would think.
Yeah, exactly.
It's like, it would be surprised, to be honest.
There's lots of room up front, you know.
Hey, Nick, is this the webcast?
Don't be back.
Oh, okay.
Yeah.
So we didn't have to can't take.
So we're getting close on 6.
See whether so let's first have to it's not common.
You know, it's
Yeah, that's great. So what do you have to do to get it? This just records it, or is it?
Yes, there's a YouTube application you download. And just plug a camera in the
Is it all free?
Yeah, it's okay. That's good. That's the whole thing.
How big is that file?
That's 340 megs per hour.
Per hour. Yeah, right.
It's such a bad idea. So you can run into a gig for 3 hours. Now, you're planning to do that.
All right. Send me a budget.
And I would have to explain.
Hey, Adrian.
Yeah, no worries. We have we just
You need to wear a mic or anything. Just
Because it wasn't as effective. So it's like, again, it was up in the middle. But if I didn't previously say it, wonderful.
Great coats. All right, I guess we'll begin. You know Howard who but today I'm wearing a different hat. That is a member of the Occupational Environmental Health Division that James Scott heads over there in the corner. I am absolutely delighted to welcome Niladri Basu, professor of McGill. He and I have a close connection. When I was at the University of Michigan as the chair of the Department of Environmental Sciences, we recruited Nil as one of our first tenure track faculty recruits. He was Canadian, and the paperwork you have to fill out to hire a Canadian is just as bad as the paperwork to hire an American here. And we jumped through every hoop, and he joined us and had a spectacular career at Michigan. He's one of those rare scientists who starts off as a sort of ecotoxicologist with some incredible work in fish and wildlife, but seamlessly moved into big human population epidemiology, gene-environment interactions, and things that are very impactful in policy. So Nil is actually also a collaborator of ours in our Element project, our early life exposures in Mexico to environmental toxicants project, a project birth cohort study that started in Mexico City with our collaborators at the National Institute of Public Health back in 1992. That's won all sorts of awards, and it's been continuously funded by NIH since then. We intersect on many different fronts, and I'm delighted he's here. One other great strength of Nil's is that he's done some groundbreaking work in Sarnia with the First Nations communities there. Maybe we'll hear about some of that today. Those combination of interests and strengths make him a particular interest for collaboration work here, because it's, as you know, the new Institute for Indigenous Health that we have created this summer. And we've already been notified by several of the chiefs that environmental issues are very high on the list of things that they would like the Institute to address. So please welcome Niladri Basu.
Great, thanks Howard for the kind opening remarks. My wife is a professor at McGill too, and this morning she commented to me, Nil, I haven't seen you so nervous in a decade in giving a talk. And she failed to realize that I was born 2 blocks from here. My father spent his whole career in the Ontario Hydro building, and that for the first 20 years of my life, my journey often involved taking the GO train from Scarborough to here. So for me, it was a real treat to take the GO train this morning and come here, a place that I've come many, many times. But then during those 20 years, I often wondered if I'd ever come to U of T for anything. And lo and behold, I finally get an invitation to speak here. So I'm thrilled to be here. And my talk today will be a journey, not of my first 20 years of life, because those are just written with things that I want to talk about, but the last 10 years. So during my 30s, and how I started off as a professor with certain biases, certain perspectives on the world, but profoundly shifted within a 10-year period. So what I thought I'd do is I'd walk you through that, and I had to come up with a title, and this is it. And I'll try to use that as a narrative for my presentation. So this is the outline. I'm going to talk very briefly on Mercury as a global pollutant. I'm going to talk about what this means to individuals and how we use guidelines to protect health. Then I'll talk more on a societal level and some of the assumptions that are being made, some of the beliefs and what this really means. And then I'll give a brief snapshot of the work that we do in Dalla. So really quickly, Mercury. It's a global pollutant. Map of the world here. Areas shaded in red are major mercury sources to the world. The thing is, once mercury is emitted into the atmosphere, it can travel long, long distances. It can stay in the air for upwards of 1 year and travel to far-off places. What we've seen in work that we've done in the Arctic, I've been working with the Arctic Monitoring Assessment Program for about 10 years. And this is back in the day when I did more work on fish and wildlife. We looked at museum specimens, we looked at archival specimens to show that levels of mercury in the Arctic have gone up about 3 to 5-fold in the last century. So the x-axis is years, and the y-axis is percent of current contributions. And looking at a diverse set of media, different organisms and people, you see that there's a consistent increase in the Arctic, which is somewhat striking. And for those of you who are familiar with the Arctic, you may know that Arctic people in Arctic ecosystems are perhaps one of the most contaminated in the world, and that's somewhat ironic. So really quickly, how is it that mercury gets from ground into people? Well, the major source of mercury is coal-fired power plants. So as an aside, that's changing right now. There's this type of mining called informal mining, which I'll talk about later, which is believed to be the major source of mercury. But let's just focus on coal for a second. Mercury is a naturally occurring element. It's bound to sulfur quite readily in certain situations. Anytime you burn coal, you also release mercury into the air. Mercury's chemistry is quite fascinating. It gets released into the air as a long-term gas. It can travel for long distances. Eventually, it might get oxidized. The oxidized form will come down onto landscapes. If it gets into waterways, that inorganic form of mercury might get biomethylated. That biomethylated methyl mercury form is important because it can readily cross biological membranes and build up food chains. In fact, the concentrations of methyl mercury in fish and in fish-eating wildlife, including fish-eating people, can be upwards of 1 million times greater than the mercury in the air or the water. So it is a chemical that, again, can traverse the world, and once in certain ecosystems, meaning aquatic ones, it can easily build up in the food chain. We care about this because methyl mercury is extremely toxic. We've known for decades that it's a potent neurotoxicant. And in the last decade, we've started to realize that methyl mercury can also be a potent risk to the cardiovascular system. There have been a number of numbers thrown out there, but estimates suggest that methyl mercury pollution, both in terms of its effects on neurodevelopment and cardiovascular systems, is costing society, North American society, on the order of billions of dollars per year. In fact, there was an estimate in the US that this chemical every single year is costing the US society $44 billion in lost productivity. These are profound numbers when you think about them. The challenge here is that mercury is mainly found in fish and seafood, but we're continuously told that fish and seafood are good for our health. And oftentimes the messages that emerge from various agencies and organizations conflict each other. So where are these guidelines coming from? There have been a number of good, strong studies in the mercury field over the years, and perhaps the most famous ones come from the Faroe Islands and the Seychelles. They've been run for decades by colleagues of mine. I know Howard knows these individuals quite well, so they're very goodly designed studies, really nice birth cohort studies that have shown, in some cases, not all cases, that early life exposures to methylmercury may matter, meaning that it may affect the neurodevelopment of the offspring. And then from that, numbers are derived. So when you look at the epidemiological data that suggests that early life exposures to methylmercury are affecting later life health outcomes, numbers are derived. And in this case, big councils have come together. So in particular, what I'm going to do is to go through the numbers that the National Research Council came up with maybe 8 years ago, 10 years ago. When they looked at all the data, they came up with a lower benchmark dose level. So this is a level of mercury in the body that they came up with, which they believe to be associated with increased risk of harm to the child. And that BMDL number they came up with was 58 micrograms per liter, 58 part per billion of methylmercury in the blood of the mother, at sort of the lower concentration. However, when you take a number like that, you need to err on the side of caution, or you want to err on the side of caution and protect all. You also want to account for the great uncertainty or variability that might exist in populations. So they used a seemingly arbitrary uncertainty factor of 10 to account for this. And so the number dropped to 5.8 part per billion. And when you look at 5.8 part per billion of mercury in the blood, and think about what that really means, again, it's coming from fish and seafood mainly. It makes most conclude that you should not eat fish or seafood more than twice per week. Okay, so the messages that came from this exercise was that we recognize that there's mercury in fish and seafood. It's not good for developing children. It's not good for the heart. We need to limit, we need to message to populations that you need to limit the consumption to less than 2 servings per week. Nutritionists look at this differently. Because in the nutritional sciences, it's been well established for a long time that fish and seafood are extremely important for the development of the heart and the brain. And when you look at guidelines from the American Heart Association and many other organizations, they're telling the public to eat fish and seafood more than twice per week. So we have two completely different messages. On one hand, messages coming mainly from a toxicology community are saying that you should limit your consumption to less than twice per week, whereas messages coming from the nutritional community are saying that you should eat fish and seafood more than twice per week. So the messages are rather confusing. So initially when I came into this more than a decade ago, I came into this from a toxicology perspective. I was trained to think as a dose that makes poison, and obviously mercury is not good whatsoever, and it's not that good. But then over the years, I started to question the process by which these numbers started to emerge. So I put question marks on a few things that I want to touch upon really briefly. One is the Faroe Islands and the Seychelles. The Faroe Islands is northwest of Scandinavia, and the Seychelles is east of Africa. Is anyone here from the Faroes or the Seychelles? No, so we're talking about very small populations of people, very genetically homogeneous, which is not that reflective of the average North American. We're also talking about two communities that eat fish and seafood in a way that none of us would ever eat fish and seafood. So in the Faroes, they're mainly eating pilot whale, and they're gorging on pilot whale once a year in these massive feasts. And pilot whale has a lot of mercury, but it has a lot of other stuff like PCBs and certain other organic chemicals. And then if you go over to the Seychelles, they're also eating a tremendous amount of fish, maybe 10 to 20 times more fish than any of us or anyone in the general public here would eat. And that fish and seafood is big fish, and it's loaded with selenium too. So we have two populations far removed from the average North American that, sure, they're getting a lot of mercury in their diet, but the types of mercury or the sources of mercury and the way that they're exposed to mercury eating fish is quite different than what we're experiencing here. Yet, it's the data from those populations that's guiding decisions here. Go down to the uncertainty factor, this tenfold uncertainty factor. Again, I mentioned that it's arbitrary. I mean, there was some thinking behind it, obviously, but it comes from a realization that people are different. So let's recognize that people are different in the way they handle mercury, so the toxicokinetics, and people are different in the way they might respond, so the toxicodynamics. So they use 3.2-fold and 3.2-fold, multiply those together, you have 10-fold. I would argue, though, now we have 21st century scientific tools that should help us to better pinpoint true uncertainty associated with this. When these uncertainty factors and the calculations were derived, they were largely not considering the beneficial effects of eating fish and seafood. And then in the end, when the messages were made, agencies did it on their own without consulting other agencies. And in fact, there's a well-documented case of the EPA and the FDA within a week of each other coming in with very distinct recommendations to the public, which this happened about a decade ago. It really caused tremendous confusion. So my tack to study mercury is, well, I feel like I use mercury because I got into research to see the world. And going through university and to grad school, I really just wanted to travel and see the world. And along the way, I started to learn of mercury. Mercury being a global pollutant, it also offers myself and my students a way to see the world. So we study mercury around the world. As Howard mentioned, my formal training isn't fish and wildlife biology, but upon joining the School of Public Health, I quickly realized that I wanted to do epidemiology. I want to study people, and I want to study people and the animals they eat, and the animals that inhabit the same ecosystems all together. I want to do this in many, many parts of the world. So we do take a global public health approach to this. And in doing so, we have a number of different cohorts that we've been working with over the years. So going vertically from the bottom to the top, we have groups that have background levels of mercury, such as us, to groups that have high levels of mercury exposure. And then going from the left to the right on the screen, we have mercury in its two main forms. I've largely talked about mercury as methyl mercury in fish, highly, highly important, but mercury can also exist in this inorganic form. Inorganic mercury is what makes up dental amalgams that some of us may have in our mouths. Inorganic mercury is a form that you might find in compact fluorescent light bulbs. It's used in mining. Inorganic mercury and organic mercury, they're both mercuries, but they act and behave very, very differently in the ecosystem, but also in the body. And oftentimes the biomarkers we study are confounding both together. So we really need to tease them apart. So in terms of background, we rely upon data sets such as the US Enhanced Canadian Health Measure studies. We have access to data sets on the order of 1,000. I'll talk about the dental cohorts in a couple minutes. Howard leads these great studies in Mexico City with the element cohort. In recent years, we've been doing more and more work in West Africa, particularly in the realm of artisanal small-scale gold mining, which I'll touch upon later. That's on the upper right, the HTM. And in the last year, upon returning to McGill, we've been involved in a couple of the Inuit health survey studies. So we have access to a cohort of about 1,000 from northern Quebec and up in the northwest in terms of doing some G-environment type studies. So what I want to do briefly now is to talk about the scientific research that we do in my lab and our focus on individuals and trying to understand variation within individuals. So the biomarkers we use for mercury come from hair, urine, and blood, and they tell us similar yet very different things. So hair is mainly methyl mercury that comes from fish or seafood. Urine is mainly inorganic mercury that's coming from, let's say, amalgams, probably the major source in the general population. And blood is mainly methyl mercury unless you have a lot of amalgam. In that case, the blood may have a fair bit of inorganic mercury in it too. However, there's tremendous inter-individual variation and uncertainty in these values. There's variation across individuals, and there's uncertainty in the way that we measure these things. So this is just an example of a couple of the numbers that are often tossed around in the field. And it complicates risk assessment because for each of these types of measures, we use a single value. So for example, this idea of a hair to blood ratio ranging from 140 to more than 600, it's legitimate, it's true, but risk assessment uses a value of 250. And in many cases, that greatly overestimates or underestimates risk. So in the end, I touched upon these uncertainty factors, and this is just some text to go along with that. And in the end, they increase the margin of safety to protect the most sensitive, but they may be insufficient or overprotective again, because they're targeted towards the toxic chemical mercury, but they're not necessarily considering that mercury is mainly coming from fish and seafood. And when you limit your exposure to mercury, you're also limiting your exposure to fish and seafood. So I'm going to walk you through a case study that we've been doing with the American Dental Association. We've been working with them for more than a decade to mainly look at mercury exposure questions and in more recent years to look at whether or not mercury might be associated with changes in blood pressure, cardiovascular outcomes. And this is just a table with a lot of data on it to just give you a snapshot of what we've been doing. So we've been working with the American Dental Association since 2003, largely to look at biomarkers of mercury exposure. They have great interest in mercury because, again, they're dentists and they're dental hygienists and dental practitioners. While traditionally in the past they were placing amalgams, nowadays people are not placing amalgams in North America, but rather these are individuals that are removing amalgams. And during the process of removing an amalgam, and an amalgam is 50% mercury by weight, we see dental professionals being exposed to mercury. So with that being said, the exposures of dental professionals to inhaled mercury now are about tenfold lower than they were maybe a decade or two ago. So there's been great changes in that community in terms of preventing exposures. Nonetheless, they still view mercury to be an important issue. It's a hypersensitive issue in the dental field, and the American Dental Association, to their credit, they have a very rich science council who fund a fair number of projects to increase their membership's understanding of the risks that may come with mercury. So they've been enlisting our help over some time. So in recent years, we've been going to their association meetings where they have great opportunities to sample hundreds, if not thousands of people. And we've been doing more focused exposure assessment studies on this population. But in recent years, we've been increasing the number of things that we can measure related to cardiovascular disease, and also in terms of genetics. So we've been doing some of our early gene environment genetic polymorphism work with this population. And in recent years, we've been looking at epigenetic type effects. So this is just an example of the type of data that we can get from these studies. On the x-axis is the calculated exposure to mercury via fish and seafood consumption as the micrograms per kilogram body weight per day. And on the y-axis is the amount of mercury in the hair. So the x-axis comes from very detailed surveys we ask of the dental practitioners in terms of the types of fish they eat, how often they eat the fish, the serving sizes, so on and so forth. And in the y-axis, we measure mercury content in the hair. And when you look at the two together in the scatter plot, there's a positive correlation. And in fact, many groups have shown this. This is not rocket science. And I will say that when you put these two together, the R-squares, the correlation coefficients are usually between 30 and 40%. So fish explain, or the surveys, the way they're designed, explain about 30 to 40% of the variance in the hair mercury. So where is that 60% coming from? And also, there's still tremendous variability in these numbers too. So we've been interested in trying to look at these types of questions and to try to better understand why there's variability, and can we do anything to improve the exposure assessments. So my interests are within toxicokinetic pathways, so systems within the body that are designed to help absorb, metabolize, distribute, eliminate chemicals like mercury. The thing is, a lot of these genes are highly polymorphic, meaning that you and I have these genes, they have an important function in our body, but you may have a form that will rapidly get rid of these chemicals, whereas I have a form that's going to keep it in my body. That's what we're starting to see. So what we did here, and this is just one example of many, we genotyped the population. And in this case, we looked at selenoprotein P, which is a protein in our body which helps move mercury around. And there are there are two major forms of it. That's indicated by the symbols, different symbols. So if we look at people that carry the major homozygote version of this, 52% of the population, and you do a scatter plot, and if you look at the regression, you get a slope-response relationship that looks like this. If you look at the remaining, those that carry the heterozygote or the minor homozygote, they have a very different, significantly different slope-response relationship. So looking at a figure like this, what you would conclude is that those that eat a lot of fish, if they carry the major homozygote, they have a lot more in their hair than those that carry the minor homozygote. So what does that mean? So in the mercury community, we've been perplexed by this, just in terms of, if there's more mercury in the hair, is that good or bad? More mercury in the hair is a function or a proxy of being exposed to more, but maybe it's not that bad because you're getting rid of it from your body. So that's the type of question that I think the community is still grappling with right now. But for the purpose of this presentation, we'll use hair mercury as a proxy of exposure. Another way to look at this is just to come up with a plausible yet different way of looking at the data. So instead of on the x-axis putting the survey information, what we did instead is estimated how much mercury an individual would be exposed to if they ate no cans of tuna per week, 2 cans of tuna per week, or 6 cans of tuna per week. There are people that will eat 6 cans of tuna per week, and there are even more people that eat 2 cans of tuna per week. So this is quite normal in a population that you get this much tuna consumption. When people are not eating any tuna, as you would expect, there's no mercury, there's limited mercury in the body, so there's no difference between those that carry the minor or the major allele of this gene. But as we start going up in exposure, we start seeing this divergence in the amount of mercury in the hair. So again, there's a significant difference in this hypothetical example using real data, where people that are exposed to a lot of mercury via tuna consumption have a lot more mercury in their hair. Okay, so this is just, I think, a classic example of a gene environment type study where you need to look at the genetics to unmask the effect.
Why is it non-linear? I mean, 2 cans and all of a sudden this huge jump with 6 cans.
Yeah, we're not sure right now. I mean, it's just a hypothetical example. If we put in 3, 4, 5 cans, maybe we would see something else. But
2 cans is what, 4 on the y-axis there, and 6 cans is only 7.
yes,
I mean, is that the scale or?
it is. Yeah, no, sorry, that's the actual amount of mercury. Yeah, so that's the amount of mercury that we're measuring in their hair.
So maybe there's some sort of saturation in kinetics.
Possibly, yeah. But this is just one gene showing something like this. We have dozens of other genes that show something like this too. So it's a matter of trying to string this all together to see at a pathway level how certain combinations of genes may better predict exposure. So we've been able to, I mean, we wrote a review paper on this last year. Our group has been asking this question. There are 2 other groups around the world which, around the same time, we were uncovering very similar results, which is always reassuring. So for those of you that study genetics, the sexy thing to do is to look at whole genomes. And that's just not in my mental capacity. I look at a few genes at a time that I understand. And same with these other groups too. We pick these genes because we know fully well that these genes are very important in the movement of mercury in the body, and so did other groups too. And it's reassuring to see that there are genes or polymorphisms in genes in the glutathione system, the metallothionein system, the sphenoprotein system, or even just transport systems that other people besides us have been studying, and they've been seeing that polymorphisms in certain genes in these pathways are exposed to and can help predict biomarker levels, both going up and down, and both for the organic mercury and the inorganic mercury. So the table here is taken from our table, just showing the types of SNPs that are consistently being shown to be associated with changes. So we concluded in the paper that these types of findings are beginning to increase our understanding of the mechanisms of action of mercury, and in doing so, we hope that they can help start identifying candidate biomarkers, maybe even susceptible groups or life stages, and ultimately lead to more accurate risk assessments and decision making. And we're starting to see this now in the European Union. There has been a recent policy report placed that's under review right now, which is putting greater emphasis on trying to pull in genetic information to reduce those uncertainty factors. I want to shift cases to one not of genetics, but one just of how the gut works. And this is something new in my group. I have a very keen PhD student who wanted to take on this question. I think it's an important question to ask. So on the left-hand side is a list going from left to right of the main fish species consumed in the North American diet. So salmon is number 1, followed by white tuna, shrimp, cod, so on and so forth. They contribute different amounts of mercury into the body. So most of the mercury that's coming in at a population level here is coming from tuna. We assume that once you eat tuna, so once you take that piece of sushi and you put it in your mouth and it goes in your gut, that 100% of that mercury is absorbed into the bloodstream. There's preliminary data coming from Europe and elsewhere to suggest that this is not the case. So again, our risk assessments make an assumption that whatever is in those surveys, when you put it into the risk assessments, that you assume 100% bioavailability. That's really not the case, as we're starting to see. So this is the available literature right now. How much mercury is bioaccessible, so coming from the fish into the intestines, and how much is bioavailable, crossing the intestinal epithelial into the bloodstream. And on the first column, again, are those top 10 most consumed seafood in North America. And in the next two columns is the data that's available on percent bioaccessibility, percent bioavailability. And the take-home message here is that we know nothing. We really don't have this knowledge. We're assuming that's 100%, but we don't have the data. And geez, the data that does exist shows that it's not 100%. So if you look at tuna, a couple of studies out of Europe that have looked at tuna have shown that the bioaccessibility is between 9 and 95%. And that's not even counting for the bioavailability. So bioaccessibility is talking about how much mercury is sort of getting sucked out of that tuna in the intestines. And then next you want to know whether or not that mercury is actually getting into the bloodstream. So we don't have that data. I don't think it's 100%. So this is some preliminary data from one of my grad students. We've gotten to the bioaccessibility part. This is a sample size of 3 only. She's been able to increase the sample size and it's rather consistent. And again, what we do see is that, yes, that for the most part, there are a number of fish and seafood species that are commonly consumed here in North America for which the bioaccessibility is not 100%. The next thing to do is to see how this then becomes bioavailable to the individual. This is all in vitro data done in a test tube. The nice thing that we can do is we can take our rather rigorous studies with dental populations where we have detailed surveys. We have a couple of other data sets with very detailed surveys and start adjusting these as adjustment factors in the risk assessment. So again, that part of the risk assessment where we assume 100% bioavailability, we can start putting in fish-specific data. And we're hoping that by doing that, we can make the exposure assessments more precise.
So you're simulating the gastric acid environment, or
Exactly.
pretty much?
Yeah. Yeah.
And is there is the uptake from the gut through like NRAM2 or one of the other?
There are a couple of organic anion transporters that are involved here that will take mercury into the bloodstream. Again, these are also transporters that we seem to be polymorphic. So here's where we want to start marrying some of the gene polymorphism data with some of the in vitro data on bioavailability and feed it all into these epidemiology studies that we do have to hopefully get better predictions between exposure and the biomarker.
And is there competitive inhibition based on other dietary elements?
There are limited studies to suggest yes. Yeah. So we can start. So once we have the in vitro system up and running, which I'm just about there, the idea is to throw things such as selenium in there. Or there's been a lot of discussion about fruits or fibrous materials. So we can start playing around with the in vitro system, but we also want to do it in a way that the data are applicable to the epidemiology. So we can link it to the epidemiology. So as a brief mid-talk summary, so all seafood are contaminated with mercury, but seafood are excellent for health. We know well that there's no safe level of mercury, so we need to protect populations against this. We use science to inform the guidelines to make decisions. However, there is great uncertainty and great variability in science, which has been challenging the guidelines and decisions. And really, I think mercury up until now, the onus has been on the individuals to balance the risks and benefits and make decisions whether or not they eat fish or not. And we
Just a question, no level of mercury is safe. That doesn't strike me as being a particularly scientific statement.
Yeah.
So, I mean, I take it what you mean is we've been unable to find a threshold for effect.
It depends on what level you're looking at. So even the most minuscule amounts of mercury, when you put them into a cell, will cause some kind of damage. And whether or not that damage can translate itself up into something meaningful for the individual is yet to be determined. But with that being said, what I mean there is that mercury has no beneficial function in any organism.
Well, I mean, there are lots of toxins that don't have beneficial function.
Mhm.
That doesn't mean that there is no threshold for effect. I mean, the benchmark dose assessment you did early on was
based on epidemiologic studies where you are looking for the point at which you find effects.
Correct.
And I mean, I find, you know, it always troubles me when we conflate safety, which I think is a political administrative construct, with threshold dose response, which I think are true scientific concepts. And I mean, we can come at it from a molecular level, or we can come at it from an epidemiologic level. I mean, certain things like lead, again, we cannot find threshold effect. Whether that means that there is no level of lead that is, quote, safe, I would argue the contrary, because we make administrative decisions of safety all over the risk map, right? So I think it's really important that we keep these separate.
Yep.
And I mean, certainly whether we're looking at essential elements or something that is non-essential, again, is an important consideration. But I really think we need to have more precision in how we, you know, look at these concepts and talk about them.
I agree. So that's a perfect segue into part 2 of my talk, in which, well, up until now, we did the work.
You were provocateur.
Yeah, I tried my best.
Well, up until now, we were doing this work with a scientific perspective in mind. As a scientist, I have certain preconceived notions and biases that science should be done this way and this way. A lot of the work I've been doing in the last few years is at the level of communities, so getting out to communities and doing community health studies. And it's been part of that process that 2 cases especially come to mind that have profoundly shaped the way that I look at this type of work and what it means. And in fact, I'm not entirely convinced that the science is really that important overall. So let's get into part 2, and then I'll conclude with where I think we should go. So society and assumptions. So let's start at Amjinong. Is anyone here from Sarnia? Great. So, and yes, of course. She was a former student from Michigan. So this is Sarnia, Ontario, Southwest Ontario, and there is a First Nations group called the Amjinong, the Chippewas of Sarnia, that have lived there for centuries. And it's quite a place to behold, because you look at Sarnia at night, and it looks at that picture on the upper right, upper left. That's not New York City or Manhattan or Toronto. That is a dense population of industry that you can view from the backyard of this scene in the bottom, which is a powwow at Amjinong that we attended a couple of years ago. And if the land is polluted, so are people. And I think this is a perspective we hear from First Nations around the world, that if the air, water, and soil is polluted, so is the soul, and you just cannot disentangle that. So this is a picture of Sarnia from the maps. On the left-hand side is Michigan. We have the St. Clair River bisecting Canada from the US. On the right-hand side is Sarnia. Sarnia is up on the top of the city. And each of those dots and points refers to an industry that's reporting to TRI or NPRI in Canada or the US. Each of those are reporting facilities. The First Nations Reserve is right here. There's no industry on the land, but I would argue that they're surrounded by industry, they're rained on by industry. And in fact, the report that came out of Eco Justice, which I believe is a legal NGO here in Toronto, calculated that these facilities back in, I think, 2006 or 7, cumulatively released 120,000 tons of pollution into the air. So a fair amount of stuff being released in a very, very small area. What drew us to Amjinong was this observation in 2004 by the community at their annual softball tournament that, oh geez, there are 4 teams here, and 3 of the teams are all female. And 1 of the teams are all the boys. And yes, boys and girls can play on separate teams. And then it just struck them that there's been this profound shift in sex ratios in this community. They did a scientific study. They published it in Environmental Health Perspective. They got a lot of traction in news. It has not been replicated since. But nonetheless, there's a lot of believability that this community is being exposed to a lot of chemical, and it's not having a good effect on health. So there's this belief there's a magical boundary. You have a beautiful forested First Nations reserve, and then you have industry right beside it. And the community has been asking for decades for work to be done. So here's a Band Council resolution from 1977 that we asked the government to look at the issue of chemical pollution falling on our lands. And here's the one taken from our particular work. So we were called in to help the community increase their understanding just very simply of exposures. They've known for decades that they're being rained on with pollution, but they've never had the opportunity nor the help to conduct a very simple exposure assessment study. So we came in and we studied the environment and we studied the people, because again, that's the way that my lab operates, but that's also the way that First Nations think, that if the land isn't polluted, they're polluted too. So we took both an ecological perspective and an epidemiological perspective. Here is the First Nations community in red. This is just an example of mercury levels in sediment, and we see mercury everywhere as we would expect, but the highest levels were out in the community lands. So we looked in people, and this is just a snapshot, it's unpublished for certain reasons, of a couple mother-child pairs of our larger study. We had 100 people participating in the study. We looked at maybe 40 or 50 different chemicals. So this is a couple mother-child pairs. We measured mercury in their hair and their blood. This is the hair mercury data. And they all have mercury in their bodies. It's not surprising. But what's surprising is the amount. It's not that high. So that line up top is the average mercury in the average American citizen. And the levels of mercury in this population are much, much lower. And that's because of this. There are signs everywhere to be warned that the lands are contaminated, that the fish are contaminated, the fish not eat the land, that stuff. So this is reflected in the work that we found. The surveys indicated people are not eating fish and seafood because there's great concern about the safety of these products, and people are just not being exposed to mercury whatsoever. And the reasons why these signs exist comes back to the story I was telling you about guidelines. That in the 1970s and 80s, when there were these observations that there can be tremendous amounts of mercury in certain communities, especially First Nations communities that live off the land, that immediately, I think, the public health response was to advise against eating fish and seafood. What's happened since then is we've seen communities across Canada, but also worldwide, that have heeded this call. They've markedly shifted their diets from that of traditional foods to market foods. And on the community here, I'm just wondering if this is reversible or not. It seems like we have a couple generations that it will take to convince them otherwise to live back off the land, but by that time things will change completely too. Let's go over to West Africa really quickly. What this individual is doing is mining in a very informal setting. In his hand is a vial of mercury, and he's adding a couple drops of mercury into that pan because in that pan are a couple flakes of gold. And mercury has a very high affinity for gold. And in the process of getting the gold into a little nugget form that can help feed his family and get rid of the mercury, he burns it. And he burns it right in front of his face. And the amounts of mercury they're being exposed to are at least 1,000 or 10,000 times higher than any of us would ever imagine being exposed to. So when I was called into this study, as a toxicologist, I was salivating at the thought of going to a community where there's going to be tremendous exposures to mercury. And we can have a profound impact on this community by doing some really good science and making recommendations that people will follow, and the situation will improve. I was wrong. So this is how the mining occurs. This is not multinational, big-scale mining. This is informal mining that is a poverty-driven enterprise. More than 10 million people around the world mine like this. More than 100 million people live in these communities. It is really a poverty-driven enterprise. And this is how they mine. This gentleman has gone down the mine shaft maybe 10, 20 meters. And once you go down 20 meters, I only went down 2 meters, so I was not going any further. But once you go down 10, 20 meters, you can go on for hundreds of meters underground. Every time in Ghana, I read a newspaper article of these mines collapsing, people dying. There is no safety whatsoever in these mine shafts. My students know full well to not walk around these communities at night because these holes are not covered. Okay, so tremendous accidents and injuries that occur. Once that rock is brought up, guess who does the crushing? It's the women. So what you have here are the women that live in the communities doing the crushing. And by the way, those pits, so this is a home, a household. that shaft is in their backyard because whatever is underneath that ground is theirs. They don't want to share it. I mean, there is some community sense, but you want your own little mine shaft. So this is done in their backyard by their family. The women are doing this. It's extremely tiring work. Look at all the dust in the air. I mean, there are a lot of public health risks in these communities. And I came in thinking mercury was a problem. Mercury is not the problem. There are a lot of other problems here. We think mercury is just a small problem. And in fact, the greatest problem here is that the walls of this house are impregnated with mercury purposefully, because for more than a century, gold has been mined in these communities using mercury. So they look at mercury as a vehicle to get gold. Mercury has supernatural powers in West Africa and many other communities around the world. There is no way we can come in and tell them to get rid of mercury. They view it in a very, very different sense. It doesn't matter how many guidelines or recommendations we come up with, we will never convince them to get rid of mercury. There are other things we're doing since. So it was these two studies that really opened my mind up to what are we doing with the work that we're doing, and how is it actually feeding into affect change. On one hand, the data that us and others have generated have resulted in guidelines which called into question how much fish and seafood you should eat. Then you get communities in many parts of the world stopping eating traditional foods. On the other hand, we think we can come into a community like this and document high levels of exposure. We think mercury is the only issue, but there are many, many other issues. And in the end, we're probably not going to be able to get mercury out of the system anyway. So this is the ancient alchemic symbol for mercury. It's ironic, it looks like the devil. Because it's a devilish compound. On one hand, it's toxic. It's a toxic substance. But on the other hand, it's found in a number of products that are of great benefit to public health. And if we don't look at this issue of mercury in a holistic public health perspective, I think the messages and the messages that have emerged are erroneous. And in the end, how many people in the public really care about the science? People have very strong beliefs and values of whether or not to vaccinate their children, or whether or not to get dental amalgams, or what will they do if a compact fluorescent light bulb breaks in their house. Should they eat the fish or not eat the fish? I mean, these are all things that give many, many people visceral responses and visceral reactions, which are not necessarily founded in the science that we do. And unfortunately, a lot of these issues fall upon individuals to make decisions. And when we think about public health, prevention is perhaps where we can make the greatest impact. So what can we do to prevent mercury on the global level? So actually, I'll just jump to this in the interest of time. We're lucky in the world of mercury that last year, in October of last year, more than 150 countries adopted to the Minamata Convention on Mercury Pollution. So this is a global, legally binding treaty which a majority of the world has signed on to. So the map on the upper right is a country of all, a map of all the countries that have signed in blue and all the countries that have ratified in green. The US has ratified. And once 50 countries ratify, this will come into force. So things are moving really, really quick. And the whole purpose of the Minamata Convention is to recognize that for too many years, there are too many great products that have great public health benefits with mercury in them. And it's relied upon individuals to make decisions on how to deal with them. There are alternatives for many of these cases, and we just need to get mercury out of many of these products. And that's a major motivation of the convention. And if this actually works, what we hope to do is to see it go the way that the Stockholm Convention did for many persistent organic pollutants, that maybe in a decade from now, I won't be in the mercury business anymore. So this is something I've been involving myself more and more with. We had a great meeting in Vancouver last week to try to talk about implementation and how to best go about infusing some of the science into the biomonitoring. So I think in the interest of time, I wanted to just mention a couple other things. And I'm not sure how much folks are aware of the element birth cohort study that Howard Hu has been running for more than 20 years, based out of Mexico City, but it's had a profound influence on how the world manages lead. It's such a great resource, the way that it was designed, that we're now looking at mercury in this cohort and fluoride in this cohort as well too. The many strengths of the cohort, but one is that during multiple periods of pregnancies, a trimester 1, 2, or 3, surveys were conducted, samples were collected. So we can get some really, really nice, important snapshots into developmental exposure, developmental windows of vulnerability. So it's a great cohort that we've been able to work with Howard on for a number of years. The mining studies, I'm going to skip over to this one. In the last few years, I've gotten myself more interested, more excited about trying to strengthan capacity around the world. So we've received a fair number of grants from foundations and the Fogarty International Center in the US, National Institutes of Health, to set up a regional environmental and occupational health network in West Africa. And I was mentioning to Howard that in the last 6 years we've been doing this. It's followed Moore's Law in terms of doubling every single year in terms of funding and people involved, and also countries involved now too. So the whole idea is to really increase capacity in the region. Our hub is based out of the University of Ghana School of Public Health, which is doing some marvelous things in that region. And the work that we do in Africa now, when we initially started it, it was being done by University of Michigan students going there for the first 2 years. Not anymore. All the work we're doing in Africa is done by a number of postdocs and PhD students that are Ghanaian students who are getting Ghanaian degrees or spending time with our institutes here. So the work has still focused on informal mining. We have a number of projects in the electronic e-waste industry right now. Our partners are doing work on pesticides, so on and so forth. But we're really trying to focus on increasing capacity for the Ghanaian students and their mid-career professionals and senior mentors to do the work themselves. Another thing of great interest to us are blood spots, dried blood spot samples. There's a lot of discussion as to the importance of early life exposures and what that might mean on later life health outcomes. But how do we go back in time and actually understand what an individual was exposed to in utero? The great thing is that here in Ontario, Michigan, and many other places, when children are born, somewhat painfully, their heels are pricked right away, and you get a blood spot sample, which is then used to do some genetic testing, which can do some interventions immediately. A lot of places bank these samples, and there's emerging technology that us and others are using to go back into these samples to understand what kind of chemicals people may have been exposed to in utero, what kind of nutrients were people exposed to in utero. So that's something that we're spending more and more time on. Another great advantage of these blood spots is that we can use them in low and middle income countries where it's just not convenient to collect blood samples in far remote sites. So again, part of the work in West Africa, we're part of a couple population health studies in which we're trying to collect dried blood spots from the field to get a more objective index of health. So with that, I see the time is 1 o'clock, so I'll stop there. And if there are any questions, I'll take them now, or you can email me at the email address below.
Well, those of you who have to go, but that's an incredible amount of great work happened into 50 minutes. So I'll take some questions if got any.
Nice, excellent program. It's lovely to see the transitions you've been through. And can you comment a little bit about how successful you've been about changing beliefs? So just take example of something like in a First Nations community where they might be, first of all, believing that mercury is the problem, and they're so happy and welcoming when you come in. And then when you observe that actually the levels were lower than might have been expected, and that other complex issues might be at play, have you been able to see sort of impacts that relate to taking on the complex issues and not just focusing on mercury?
For myself, no. In the limited experience I've had working with First Nations community, there's great variation within the community in terms of their beliefs. All in all, the belief is that there is a strong connection between the body, soul, and the environment. So that's one deeply rooted belief which permeates across the community. But then within the community, there are certain individuals in multiple communities who are very vocal about this issue. And there are many others who wish it just never existed. And that was my experience in Amjan, which is the community I've had the most experience with. And the results we got, they didn't really satisfy anyone. Because on one hand, we didn't have the smoking gun that the activists were looking for in terms of tremendously blazing high exposure that explained everything. But on the other hand, those that are just hoping for a clean bill of health, there's still chemicals there. And yes, there are rather small concentrations, but nonetheless, the belief and the long-held belief is that even the smallest amount is not natural or normal. So I don't know if I really gave a satisfactory answer, but we've had no.
Describe the reality. I think you're not alone in that experience. And, it's a long journey we're all.
Yeah. So that in many ways, if you just think about prevention, why do we have to continuously put the onus on individuals to make these decisions when it's possible? Maybe through some policy ways to get rid of these things.
Thank you.
Any questions? Great talk, thanks. Puzzled as to why you say that none of us here consume that much fish. I mean, we have encountered clinically some folks with blood mercury as high as 200 micrograms per liter. So there are people who do eat an incredible amount of fish in Toronto and other Canadian cities. So, I mean, I was certainly taught that First Nations mercury exposure was the heaviest anywhere in Canada 30 years ago. I don't believe that anymore, given what we found in many urban centers. And the other thing is that a lot of that fish is actually clogged rather than caught. So I think there are some quite different consumption patterns within the urban Canadian and probably American population. And we do have some people with very high exposures.
Yeah, I don't disagree with you. So I think I was talking in general terms, and just in terms of population exposures. If you look at Canadian health measures data and data, they're largely showing that 5% to 95th percentile are rather lowish in terms of.
Yeah, although
there's always going to be
the irony then on the guidelines, I think, becomes that because you've got those population surveys, the results of those get used in a clinical sense to set kind of normal ranges, and those are really not reflective of people who have different dietary preferences. So we've got some Health Canada funding coming, and we will be able to look at some of those subpopulations, but I think it fits with your comment about guidelines, and ironically, some of those surveys, I think, are being misinterpreted by Health Canada and other organizations to set guideline intervention levels that are really contrary.
That's exactly where we want to go. I mean, there's this one value, 5.8 part per billion, which is intended to protect us all. I have a very hard time believing that. And the genetics data itself is showing that this is not holding true. There have been a couple of really interesting studies to take the normal EPA risk assessment, compare it between Inuit, Cree, Japanese, Caucasians, and the numbers are always wildly different. And then if you look at a couple of the gene polymorphisms that we've been focusing on, they're minor allele frequencies, so their frequencies vary tremendously across these populations. And again, this basic equation has been used to determine 100% of people in the north are at risk of mercury poisoning. But if you really look down in the details, how true is that? And I guess in the end, we need to have group-specific guidelines, but that requires a lot of resources to do.
Any other questions? Yes. I really enjoyed hearing about your experiences with ASGM in West Africa, particularly because I've studied ASGM in Ecuador, and there seems to be like a lot of similarities there, but also quite a few differences. I'm wondering, in your experience working there, what is the role that policy plays, you know, in terms of government, like top-down kind of stuff, as opposed to science and education, bottom up, like coming from within the community? What is the role that that plays in resolving that issue, in your opinion?
So the government has a profound role to play because a lot of these communities are the poorest of the poor, often neglected, completely under-resourced. With that being said, there are certain governments that recognize these communities need help. Ghana is one, for example. So they have outreach centers in each of their 10 districts, which are designed for these ASGM miners, where the miners are supposed to register. If they register, they get resources from the government. The Minamata Convention is going to help tremendously. There's millions, if not billions of dollars by donor countries that are intended to flow to low and middle income countries, principally or mainly for ASGM work, to help them better improve the situation on the ground, focusing on Mercury, but using Mercury as an entry point to take care of a lot of other public health issues. So with that being said, I think it's really going to depend upon the will of a local or a national government and the types of programs we're willing to implement to affect change in their country. It's going to vary country to country. So I give the Ghana example because I know it the best, but I know that as part of the president's task force, ASGM is amongst the top 5 or 10 issues. So the country that recognizes this to be a big, big problem, they're going to invest heavily in it.
The artisanal gold mining thing reminds me of a similar problem among lead-exposed populations. There's Alan Counters, a colleague of mine at Harvard, who studied these populations in high regions of Ecuador that were taking old car batteries filled with lead and cooking them up in their backyard, big frying pans, then taking the lead and making ceramic roof tiles for them and then marketing roof tiles. And they've been doing this for over 30 years. And it's so entrenched, in the most poverty-stricken part of Ecuador, that it's unlikely that anything is going to change for some time. But one of the curious things about it is that he did an analysis of looking at lead exposure and some of the usual neurologic and behavioral outcomes. And he found a much lower dose response than has been seen in the international studies. And looking back at some of the birth patterns of populations there, it's almost like you're looking at ecological and environmental evolutionary change already, because there's already been an impact on selecting people who are probably less vulnerable to lead's effects. I wonder if that might be also happening.
It's possible. Ultimately we'll do some more genetic studies too.
Surely. You look at population genetics and evolution and selection. The idea that will take place in a couple of 100 years of the post-industrial era, that's really pretty tough to, I think, believe quantitatively, isn't it?
But these mining where it goes back a century. Sorry.
Even a millennium, is just a blink in evolutionary terms. So, to think that it works through genetic selection.
Well, now we're talking about people with 8 to 12 children, and which ones survive.
Yeah, but most of them are dying of something else. And in infancy.
Maybe.
Yeah, so things
curious. Really curious.
There are other issues that are contributing to morbidity and mortality that are far more.
Yeah, I would think the selective pressures such as exist are really operating other things.
Sort of related theme and bringing it back to some of the work that you presented. Clearly some of the solutions, or that's not clear, but the solutions I think are along the lines that you've actually proposed, and that's getting the local capacity building, getting an economy there that people can do, that it is actually a step up that attracts people and attracts the talent and creates local solutions. So the model that you've developed is really very necessary.
What we've seen in a lot of these communities, sure, they're informal mining communities, but there's big money. And we're seeing people going into the communities with a bit of a business sense who rally around hundreds, if not thousands of miners into, you can't call it small scale anymore, it's probably medium scale, but they also understand the value of having a healthy workforce. They see the value in having efficient ways to mine the gold. So it's been interesting in the last few years going to some of these communities and seeing change before your eyes. One great example that we have is that there's this community in remote Ghana which sold their lands to a Canadian mining company, because Canadian mining companies dominate these markets, but it's being run by Chinese. And it's just a great example of like globalization in action in this small remote part of Africa. And every year you go back and you see this thing just balloon in size. So it's gone from an informal operation to a massive entity with an address on West Hastings Street run by Chinese. Just amazing.
So are the people benefiting? Are they getting healthier?
Well, we don't know if they're getting healthier, but there's development in the area. There are better roads, there are clinics, there are nicer schools, and in the end, people are likely to benefit from that.
All right, well, let's thank